Bonjour et merci d'être venu ici. My name is Sophia Anner. I'm really excited to practice my high school French here in Montreal. The rest of the talk is in English. I am a PhD candidate at the University of Louisville, and today I will be telling you about courtship in Rhagoletis palmanella. But before I start, I want to thank the people who have supported me in my research so far. So my incredible advisor, Alicia Lackey, my lab mates, Shayna and Olivia, and the undergraduates who played a really big role in this research, Rowan, Brianna, and Naomi. So one of the primary goals of evolutionary biology is to understand patterns of variation and diversification, as evidenced by some of the incredible talks that we have seen and will continue to see in this conference. One of the facets that I am really interested in is how plant-feeding insects are contributing to this diversity. So phytophagists or plant-feeding insects drive diversity because they have very specific adaptations to their host plants. So as there are changes in a host plant, that can lead to changes in the insect. In these three examples here, a shift from an existing host plant onto a novel host plant either initiated or at least contributed to a speciation event. And so multiply this by a couple thousand or million, and you have really high diversity in plant-insect relationships. So this context brings me to my motivating questions for my overall research. How do host use, and then as well as just in general divergent ecology, influence patterns of variation at the phenotypic and genomic level? Going a little bit further, I'm interested in variation that is relevant for sexual selection and sexual isolation. So I'm focusing in on courtship and communication. And then finally, I would like to mechanistically be able to connect courtship behavior to sexual isolation that is seen between closely related but divergent species. So to answer all of these questions, which is my entire dissertation, I need a system that has sets of sister species that vary in their host use. So ideally, I would have one set of species in which each species is infesting a different host, and then I would have another set where they all use the same host. I can compare variation between these two groups, and that will help clarify the purpose or the function of host use. Fortunately for me, the Rhagoletis genus exhibits broad variation in host use across the genus. So you can see in the example with the black and white fly, each of the species, which is denoted by the arrows, is infesting a host fruit in a different plant genera. Whereas the other fly that is black, brown, and orange, each of the species is all infesting the same host plant generally. So they're all infesting walnuts in the genus Juglans. You may be familiar with Rhagoletis palmanella. It is a classic example of divergence following a host shift with ongoing gene flow, in which the ancestral hawthorn-infesting fly made a switch onto apple when apples were domesticated in the United States and underwent further divergence and have generated different host races. In my research, I'm interested in looking at variation within and between many species, so I will be expanding across both of these species groups. And recall that I am interested in communication and courtship, and so what do we already know about courtship in this system? We know that males and females emerge at the same time, and we know that they mate multiply. Both the males and females mate on average once a day, sometimes up to like 8 or 10. And we know that the flies perform a wing display when they interact. So in the image on the top, the fly has her wings at rest, resting along her abdomen. When they interact, they pull their wings forward and present their wings to the other fly or whatever they're interacting with. This has been proposed to be for predation avoidance. So in an experiment, researchers found that when flies perform the wing display, they are eaten less by their jumping spider predators than if they don't perform the display or if they have the pattern obliterated. So the insinuation here is that the wing patterning on the flies is mimicking a conspecific jumping spider, and therefore the spiders do not predate upon the flies as much. However, it is notable that the species vary in these wing characteristics, and so there's variation in the pattern, size, and shape. So overall, despite some descriptions of displays in courtship, most researchers are concluding that Rhagoletis males force copulation after minimal interactions with females. As one group of researchers said, the wing markings remain lovely and mysterious. And this has been a point of inspiration for me because I think that The courtship display and the wing markings deserve a closer look. So going back to one of my larger motivating questions, what is generating variation in communication and courtship, I first need to answer what traits and behaviors are important for courtship and how mating decisions are made. So this is the context of my first chapter and what I will be presenting today. To do this, I focused on just one population of hawthorn-infesting Rhagoletis pomonella collected in Detroit, Michigan in 2021 and then reared to adulthood for behavioral trials in summer of 2022. I did no choice trials with just one male and one female. I had 28 of them, and then I video recorded them and analyzed them in Boris, which is a hand annotating ethogram software. I measured wing display duration, and then I also measured metrics of the copulation that I'm interested in. So the latency, the success, and the duration. Success is measured if they copulated within the 30 minutes that I gave them. And so if they did not, then that would be a fail. And the latency for fails is 30 minutes. I will attempt to show you some videos that are working. Yes? Okay, so this is the female is displaying at the male who is just standing at the edge of the box. She's displaying her wings. The male is walking away. He's not very interested. And then she turns around. Here is another video. Again, this is the female displaying. She's doing something that I saw very commonly, where she walks in a little arc and displays her wings. It's often reciprocated by the male. In this instance, the male is watching and decides to copulate with the females. I want to define a few terms that I'm going to use for the rest of my talk. Directed display effort is the duration of wing display at the other fly divided by the latency. And so the reason for this is that the latency varied between the trials, and I wanted to have a standardized metric of effort to compare between the trials. But I also describe a total display effort, and this is the total duration of wing display divided by latency to copulation. This is because the flies didn't only display at each other. Sometimes they displayed at the fruit stimulus that was in there, sometimes they displayed at the wall or seemingly nothing, and so I wanted to track this as well so that I could differentiate between directed and total and have a metric for overall effort. In addition to these behavioral metrics, I measured tibia length, which is a proxy for body size in this system, wing pigment and shape, and then fecundity. So the females and males were frozen after the end of the trials, and I dissected the eggs and counted them for a metric of fecundity. For my first question, how do males and females differ? I did a simple generalized linear model with the trait of interest as explained by sex. And then for my other questions that are slightly more complicated, I had many potential predictors. I wanted to look at for both the males and the females separately, size, age, display effort, pigment, and then fecundity just for the females. And so I employed an AICC model selection process in order to best answer my questions. So what I found is that there is sexual dimorphism, which has been described before. We see that the females are much larger than the males, at least on fly size. And then we also see that the females have more wing pigment. So note that this is not due to the size of the wing. This is the percent of the wing that is pigmented. It's greater in the females than in the males. I find that females and males invest similar effort in their display. So on the y-axis here, you have the total display effort, and both the males and females on average are spending about 16% of the latency performing the wing display. When you look at directed effort, though, we see that the males are spending more effort than the females displaying directly, and so they may be engaging in courtship more directly or trying to get attention more directly than the females are. I wanted to look into this a little bit closer to see what might be explaining this male display effort. Is it an honest signal of something, perhaps? And I found that male display remained consistent irrespective of the male or female traits. However, when I looked at female display, I found that it was influenced by male traits. I found that females display less when the males are older, so there may be a fitness benefit to the males in being younger. And I found that the females display more when the males display more. This was a really exciting result for me to see because it indicates there might be some sort of reciprocity or mutual signaling. The flies are communicating to each other in a way that when they do it more, the response is to do it more. In looking at other characteristics, I found that the latency to copulation was shorter when females have more eggs. So there's some variation in female receptivity that I am seeing as well in these trials. There's also really high variation in the number of eggs that a female has, all the way from just above 0 to one female had 80 eggs. I found that none of the traits explained copulation, duration, or success. So this is what I was primarily interested in to evaluate sexual selection, and I didn't see this. What I did see, though, is really high variation in copulation duration. Some of the trials, it was less than 30 minutes. The average was 68 minutes, which people are always very surprised about, and they went all the way up to 2 hours, which is actually my artificial cutoff point because I ran out of room on the SD card to continue to watch them. And I found that even though none of them explained success, I wanted to take a little further look into this trait that I find really interesting, the directed display effort. And you can see, first off, that there were only 4 trials in which the flies didn't copulate. Within this population, there was very high mating rates. And you can see, though it wasn't statistically significant, that there is an association with minimal display effort and no copulation. I'm in the process of looking into this further. I'm employing a sequence analysis using the package Traminer, which will compare more complex attributes of the sequences to each other. So here I'm showing you just examples from 4 trials, female row on the top, male on the bottom, and every blue bar is when they're performing a directed display, so facing the other fly. And again, we see really high variation here. So in this example that I'm highlighting, there is essentially no behavior. The female does one little wing display and then they're copulating. That's the black. And compared to this trial, where the female and the male are both performing wing displays, often at the same time, which means they are directly facing each other, performing displays at each other, and then copulating. So I'm hoping to look into this further in my future research. In conclusion, females are larger and have more pigment. Both males and females perform wing display during courtship, which is an especially exciting result because in almost none of the existing literature does it even mention that females perform a display, let alone as much as the males. And so I think this merits a closer look into this behavior that has been neglected before. Females are adjusting their decisions based on male age, effort, and fecundity, and males display irrespective of traits. And these results are laying the groundwork for my future work, looking at how courtship varies within and between species, and then untangling what might explain this variation. And finally, I'm excited to announce that this has been funded by the USDA NIFA Predoctoral Fellowship, and so I will be able to look at these really broad comparisons in the next couple of years. Thank you so much, and I'll take any questions. Yeah. Yeah, so I looked at everything separately. I analyzed way more than what I showed here, and I decided that this was the best metric because what I'm interested in comparing is between the trials. If you're looking just at the total duration, it's like completely driven by the latency to copulation rather than the traits of the flies themselves, which is what I'm interested in. But I do have that separate set of data, and it's something I might consider in my future trials, especially if the latency wasn't so variable. Yeah.
So do you have an explanation, or maybe I've lost it, for the fact that there is a negative correlation between the number of eggs and the courtship duration?
Yeah, so the question was if I have an explanation for the negative correlation between the number of eggs, and then it wasn't duration, it was the latency to copulation. Yeah, so I don't exactly know why. It may be an indication, and this is just off the top of my thinking, not necessarily validated, that when females have more eggs, they are more receptive, they're more ready to mate, and that might be one explanation for why. Thanks so much.
Okay, cool. So my name is Jessie. I'm also super stoked to have a chance to practice my French, so if you want to discuss this afterward, either in English or in French, please find me. Today I'm going to talk about some work that I'm doing with my colleagues Mark B. and Kim Hoke at Minnesota and Colorado State, respectively. So, if this button works, let's find out. Okay. So, across animal taxa, we have a lot of systems in which a signaler produces an advertisement signal that functions to attract receivers. Typically, signalers are male, typically receivers are female. Receivers impose selection through mate choice, and that response can take some different forms, right? In some systems, receivers might also produce their own signals in response. In some systems, like in these jumping spiders, maybe a male produces a signal and the female response looks like either attacking or copulating with the male. In a lot of acoustically communicating animals, including the ones we're going to talk about today, receivers do phonotaxis, which is movement towards sound, and that's the behavior that we're going to take advantage of today. So I work on this organism. This is the diploid gray tree frog, or Hyla chrysosceles, and they live pretty broadly across the eastern United States and southeastern Canada, so it might be a familiar organism to you, depending. And in the summer, males gather in ponds in huge aggregations, and they each produce advertisement signals hoping to attract a female. I'm going to try and play this sound so you can hear what it's like. Okay, so it's a simple pulsed advertisement signal. You might think it sounds kind of like a croak. And so the cool thing about this organism is that we know a whole lot about how, the slide doesn't want to advance. Okay, hang on. No, I don't want to do any of those things. There's like an audio driver problem, maybe. Okay, I'm going to start it over, and we're going to move past the slide with the audio. And okay. So I'm just going to keep talking while we do whatever this is about. So the super cool thing about this frog is that we have studied a whole lot about how its advertisement calls work and how receivers exert selection. Does that work? Yes. Yeah, we can see it now. Thank you. Okay. And all those decades of work have produced one thing that's extremely clear, which is the thing that frogs care about the most is call rate. They care about call rate almost exclusively. If you're giving them a bunch of different options, the first thing that they will choose upon is call rate. And so a male is producing a series of calls over a period of time, and what you might notice about the idea of a rate is that you have to estimate a rate from a series of repeated things, right? You can't get a rate from hearing one signal one time. So this male is out here, he's producing calls over and over again. And what you might notice from this waveform, so this is amplitude over time, so each one of these black bars is a little pulse of sound that's a single call. What you might notice is that these are very irregularly spaced. And this turns out to be important in a way that we didn't really recognize until pretty recently. Signals are intrinsically variable. So this male is producing this call over and over again, but it's not the same every time. So we thought within individual variation in call rate might be important, and here's a little illustration about why. So on the top here we have a waveform, so again, each one of these black bars is a single call over time. And on the bottom, for this 3 minutes of calling, I have plotted this male's instantaneous call rate under each one of these black bars. So each circle now is representing a single call. The dashed line is this male's average call rate over the 3 minute period. And you can say that over short periods of time, he's varying wildly around his own mean. Now, you might wonder how long females listen before making a choice, and our best data suggests that they spend about 2 minutes making a mating decision in the field. That's the green window. So it really matters a lot which 2 minutes they sample, because if they show up and they sample this 2 minutes, they get a relatively favorable idea of how attractive this male is. They're going to overestimate what his mean call rate is. But if they show up less than 1 minute later, they're going to drastically underestimate what this male's average call rate is. Okay? So a few years ago, we did this experiment trying to understand how this within individual variation might hide the between individual variation that receivers are trying to make decisions using, right? So now across this front axis, we have the mean call rate. This is the call rate of an alternative stimulus when the choice they're making is between a totally average standard call and one that has a call rate somewhere on this axis. On the left-hand axis, there's average coefficient of variation. So toward the back of the plot, we have perfectly consistent, robotic, machine-like calls. And on the front, we have much more variable calls. So as we move toward us, we're seeing that this preference function, the probability that female chooses a particular call, is flattening out. So at the back of the surface, we see this nearly linear preference for faster calling that we expect all the time in these tree frogs, but they're unable to do that as the calls get more variable. The next question I had was, Do the females actually discriminate on the basis of the variation itself? All things being equal, are they choosing based on the coefficient of variation specifically? And that's what this plot is. So on the x-axis now we have the variation in call rate, and on the y-axis is the probability of being chosen. And so females discriminate very heavily against variable males unless they're prevented from doing so by a whole lot of noise. Okay, which brings us to the present study. Our aim was to find out basically how much does this matter in the world when we're talking about how receivers exert multivariate selection on males in their calls. Can they do this in noisy, LEC-like environments or not? So we set out to measure multivariate selection on call rate and within individual variation in call rate in choice contexts that range from 2 choices to 8 choices. Now to do that, we needed independent male traits. So call rate and coefficient of variation in call rate tend to be correlated in nature such that males that call fast also call consistently. So we want to break up those natural variations. And we also needed a measure of relative fitness to measure the fitness of these hypothetical male phenotypes that would work across our choice contexts. And this is what we decided to do. We decided to generate hypothetical male phenotypes by drawing values from normal distributions. So we started with one normal distribution for each of the traits of interest, one for call rate, one for within individual variation. For each hypothetical male, we're drawing at random from somewhere in this distribution. So for example, this male might have a call rate a little bit below mean and a standard deviation a little bit above, and that gives us 2 independently chosen values that we can use to create a distribution that represents a very specific male, right? Now we have this single hypothetic male. Every time we would like to create an audio stimulus in this track, for the trial, we're gonna draw at random from this normal distribution, okay? So now we're thinking about males not as individual static trait values, but as little distributions of traits. And we end up with audio stimuli that could look something like this. They might be very, very consistent. They might be quite inconsistent. But then we can ask, how do females exert selection on them? We can measure female preferences using a very simple phonotaxis test. So if we're looking over the top of a 2-meter in diameter arena, the frog is in the middle, we can give her a 2-choice test, or a 4-choice test, or an 8-choice test of 8 hypothetical males that are uniquely assigned to her. And we repeated each 2-choice trial 1 time. Each 4-choice trial was done twice, and each 8-choice trial was done 4 times. That sounds kind of strange, but the idea behind that, the rationale, was that this could give us a relative fitness score that would be bounded by 0 and 0.5 across all of those choice contexts, so we could compare apples to apples and really think about how the selection acted on those unique hypothetical males in different contexts. Our sample was 125 female tree frogs. We tested each of them 12 times. So this is a 1,500 choice test data set where we measured selection on 512 unique male phenotypes. And here's some results. So we're going to look at this in 2 dimensions first, and then we'll look at it in 3 dimensions. It'll be easier to think about. So here on the x-axis we have call rate, and the y-axis is the relative fitness. Again, this is bounded by 0 and 0.5. And so we find our very familiar nearly linear selection for faster call rates, which is comforting, because if we found anything other than that, we'd be really confused right now. Okay, when we look at forced choice tests, though, that preference function flattens out. So considerably weaker selection on call rate once we get up to 4 choices. Remember that 4 choices in the context of a frog lek is not very many frogs, right? Okay, when we get up to 8 choices, again, we see a substantial weakening of that effect, so that the relative fitness of a single male is much less easily predicted by call rate. And I just want to point out to you these 2 points in the top right. There were exactly 2 females, females in the whole data set that chose the same male all 4 times in the 8-choice trials. And I'm going to come back to that later, but those are those 2 females. Okay, so for coefficient of variation, on the x-axis now we have coefficient of variation, so 0 means perfectly consistent, and higher than that is more variable. We see directional selection, females prefer consistent males. That's consistent with what I showed you before, so great. When we look at 4-choice tests, that selection is considerably weaker, and by the time we get to 8-choice tests, it's weaker still. Okay, so those are what the effects look like in 2 dimensions. In 3 dimensions, they look like this. So in the top right here, You can see that the fittest males in the two-choice tests have fast call rates and low coefficients of variation as expected. But when we think about this fitness surface that we measure when we look at 4 choices and 8 choices, overall, there's much less selection happening there, right? To kind of wrap my head around what that means, I expressed it in fold reduction of selection relative to two choices. So for call rate, 2.7 times less strong in the 4-choice test and 3.7 times less strong in the 8-choice. Coefficient of variation, 3.6 times less strong at 4 and 4.2 in 8. Right, so it's much, much weaker than we measure in our standard bread and butter 2-choice test that we have learned so much from over the years, right? And what I think is really interesting about this is we find that selection is weaker in these complex environments. Our goal was to approximate something somewhat more like the lek that animals are making these decisions in. We've only measured selection on 2 traits, which is many fewer traits than they normally are making decisions based on in nature. They are capable of assessing up to 5 things at a time, we've demonstrated, and others have demonstrated. And so their selection is acting at the level of the whole phenotype, and they're taking into account a bunch of stuff, right? So if we extrapolate this any further and we try to imagine how receivers are deciding amongst males in this even more crowded, even more noisy environment, I wonder how strong the sexual selection can be. It seems like these crowded environments where males vary in multiple dimensions at the same time are really leveling the playing field for unattractive males, which is great news for them if you're an unattractive frog, right? It sucks if you're an attractive frog, though, because all the effort, the metabolic energy you're putting into calling quickly and consistently is not paying off necessarily. It seems that females really are capable of using both call rate and coefficient of variation to make these decisions, and they're exerting multivariate selection on these traits, which is cool. But very few males were chosen all 4 times in 8-choice trials, which makes me wonder what is causing all the variation within females in their behavior. If we give them the same test 4 different times within a couple of hours, why is it that they do different stuff on different iterations? It also makes me wonder, is there an ideal time that receivers should listen before making a choice, right? If we know that this within individual variation can really bias your estimate of the traits, then is there an ideal time to listen? I want to thank the Bee and Hoke labs. They've been super helpful, including Lata and Katie. Ruth Shaw, who's here, thank you, Ruth, for helpful discussions about the stats, and my undergrads from UTK in 2023. And just real quick, we are founding an animal behavior center at UT Knoxville, and we're going to be hosting a local animal behavior meeting. So if you're in the southeastern United States and you'd like more information, scan the QR code and give us your email address. And if there's any time while we change over, I'll take questions.
Hello everyone, so my name is Camille Thomas-Bulle. I'm a postdoc with Erica Larson and Doug Emlin. And I'm going to show you today and present you my work about the genetic basis of horn size reduction in island population of Japanese rhinoceros beetles. So sexually selected weapons like antlers, horn, tusk often diverge more rapidly and dramatically than other body parts, and this suggests a history of strong and likely divergent selection toward larger size, leading to some extreme forms, size, and shapes. And weapons typically have 2 functions during fights. They act as a signal of fighting ability and as tools of battles. And in this context, larger weapons usually have several advantages because larger structures are easier to see and assess. Males do better at deterring their opponent before they engage in a fight. Longer weapons allow male to touch, strike, grab, flip their opponent before their rival can do the same, and those males are also more likely to win the fight, as the development of larger structure implies that also the development of larger muscle mass, and so added strength. And so to study that, my model system are the Japanese rhinoceros beetles. You can see here 2 males. This is a video of a fight. Each male is trying to flip the opponent off the tree trunk. And you can see a female feeding on sap on the top right of your screen, a hornless female. And so in this species, finding success can be predicted by the relative body and horn size difference between the two males. And we observed that bigger males have a higher reproductive success than smaller males. And so every night, the same dance starts again. Adults come down from the tree canopy to feed on sap flowing out of tree holes carved by other animals, and that's important. And those sap flows are rare and quite valuable because mating occurs when females are feeding on the sap of those trees. And so males, guarding males are producing aggregating pheromones to attract females and males as a side effect. And while they are fighting, the female are feeding, and then the winner male will mate with the females. So this species, Tripoxylus dichotomous, is distributed all across Asia, ranging from northern Japan to southern China. And they have been described based on their geographic location and morphology. And what you can see here is that the most variable trait here is the horn size in the male. And it goes from pretty small horn males, like in Kanemarai, the pink square, to thick longhorn, like the bright red square of the Chinese population. And so this seems to be, it seems to be that there are consistent population differences in relative horn lengths. And so the broad question that I'm addressing here and during my postdoc is what explained the variation in horn size between populations? And so to do that, I use mostly genomic, population genetics, and transcriptomic tools. And so the first question is, what is the phylogeographic history of the species, and how does the horn size vary across the phylogeny? And this work has been done by me and a collaborator, Jessie Weber, that I saw in the room somewhere. And those are the combined results of our beast and admixture analysis. And they support a deep ancient split between the north and the south clade. And we identify 9 distinct genetic populations. And it seems to be that the horn started small at first, and then in each clade, the horn got long during, right after the split. But you can see here that after this dramatic increase in size, there are 3 populations, and you can see them in the little square with the allometry. Population of Goto, so the pink one, the blue, light blue clade of the 3 islands of Kushimaru, Yakushima, and Tanegashima, and the dark blue island of Okinawa. In those islands, the beetle evolved a smaller, shorter horn. And so to understand when the changes occur during the development and what keys are making the difference in the horn formation, we use transcriptomic to follow gene expression during larval development, comparing expression profiles in long and short horn populations. And so the species has a 1-year-long life cycle. And the horn is formed during the pre-pupal stage and lasts about 11 days. And Japanese rhinoceros beetles have been well studied in Evo-Devo, and so we know that the gene cascade of the horn formation is well known, and it has been shown that this trait is nutrition and insulin sensitive. So during the development, at the pre-pupal stage, the horn cells proliferate under the larval head cap and folds like an accordion, forming discs that are called imaginal discs, and those discs unfold during the pupal molt. After the pupal molt, and so we dissected males, larvae prior to pupation across developmental timelines spanning early, mid, and late development. And so from now on, the longhorn population are color-coded in green, while the shorthorn population are color-coded in yellow, sorry. And I'm going to refer to them by the island that they were collected from. So we chose beetles from 3 populations, individuals from the mainland, the main island of Japan, Honshu, the big island, where beetles have a long horn, and 2 small islands where the horn is short, so the nearby island of Kushinobaru, I don't know if you see, anyway, and the distant island of Okinawa. And so what we found is that there are about 11,000 genes that are expressed in our data set. It seems that the gene expression profiles cluster largely by developmental stage, with greatest difference between early and late stages. But we also found that within developmental stages, there is some clustering by population. And here I'm going to show you expression profiles, and the number here are the number of differentially expressed genes that are either expressed at one stage of the development, so early, mid, or late, or at 2 stages, or during the entire development, the bottom line. And so there are more differentially expressed genes when populations are more divergent, so in between Honshu and Okinawa than in between Honshu and Kashinawaru. But we observed globally the same pattern of differential expression, that most of the genes that are differentially expressed are it's in the early phase of the development. We found 25 genes and about 400 genes that are differentially expressed across the entire development of the horn and the prepupal stage. And when we look at the function of those genes, most of them are coding for cell division and tissue proliferation, which was what we were expecting, but it's a nice demonstration. And so I was wondering if those differentially expressed genes could be under positive selection in long and short horn population. So we use pulse-seq data on beetles collected in Honshu, so longhorn population, and Yakushima, which is close from the first island that we collected from. And we found very low average differentiation with the average FST of 0.1, but identified some FST peak. And the function of the gene in those peaks are related to metabolic detoxification, wood digestion for one, and immunity for the other one. And surprisingly, those results are similar to what a Chinese team has found looking at differential expression of gut tissue of beetle larvae fed with different tree species. And so maybe in this case that it means that ecological adaptation to different tree species for the larvae or the adults could be a driver of the differentiation between mainland longhorn population and small island shorthorn population. And to support this hypothesis, I'm going to present here the work of a former master student of the Emlen Lab, Jill Del Sol. She has done field work. She's done field work to measure selection acting on this horn and measure the male mating success. And what she found was striking ecological differences. So in Honshu, beetles congregate on a small number of feeding territories, and they feed on oak trees, and they can't carve the bark by themselves. They rely on other animals to carve the bark and then access the sap. Whereas in Taiwan, beetles shifted to feed on the sap of ash trees, and the bark is way softer, and so they can carve it by themselves. And so the consequences is that there is an increase of abundance of feeding territories in Taiwan in shorthorn population, and there's a wide distribution of those territories, and this resulted in a lower probability that any particular feeding territory would be visited by a female. And so it means that this simple shift in relative abundance and dispersion of feeding territories limits the reproductive benefits of guarding males in those in those islands. And this may have relaxed selection on the male horn length and has contributed to the evolution of relatively shorter horns. And that's what she showed here in those fitness landscape. In Honshu, large male with long horn not only won fight, but also were the most successful at mating with females. But in contrast, in Taiwan, reproductive success was more uniformly distributed across the weapon and the body size spectrum. And the most successful males were not necessarily the largest or the longest horned And so small males in those islands were just like happened to be lucky to have a female visiting their territory. So that's where I stand now. So what explains the variation in horn size in between populations? So we found an early split between the population in 9 distinct populations. The horn length increased dramatically 2 times and was followed by 3 subsequent horn size reduction. There was a conservation in the gene expression profile during horn development, and the expression divergence between the horn type is greater early in the development. Looking at population differentiation, there's a low level of divergence between closely related population or subspecies, and genes within FSTP are coding for immunity and wood digestion. And it seems that maybe the driver of the horn size diversity could be caused by beetle density and force of competition, relative abundance of feeding sites, and the tree species. With that, I want to thank my lab, the Emlyn lab, the Larson lab, and my co-author and collaborator, and you for listening at me. Thank you. Yeah.
So those are great talks. I'm curious if there's a difference between islands and then the mainland with predator densities.
So the question was, is there a difference between predator density in between mainland and islands? So they have, they live for about a month as adults, but the predation is really high. I'd say there's more predation, at least in Honshu, than in some small islands. They are mostly eaten by raccoon dogs, but I don't know exactly. What we measure is that in Honshu, they have an average lifespan of 4 days as adults before they get eaten. But I don't know in small islands. That's something that we could try to measure.
Is the horn like impalatable?
Not really. Yeah, it's, yeah.